Greetings, and thank you for joining us tonight. We're about to explore a realm hidden from view, a dimension so minute it exists in the spaces between sand particles, within droplets clinging to moss, and deep inside frozen fractures. This is the domain of the tardigrade, often called the water bear. These organisms are so minuscule that observing them requires magnification, yet their capacity to withstand extremes surpasses nearly every other living thing on our planet. Tardigrades have earned descriptions like indestructible, immortal, and unkillable. They can become completely desiccated for years on end, endure freezing near the coldest temperatures physics allows, withstand pressures that would flatten most creatures, and even remain viable in the void beyond our atmosphere. Four beings that resemble plump, eight-limbed miniature bears, their toughness ranks among nature's greatest achievements. Tonight, we'll journey through 100 remarkable facts about these extraordinary animals. Some will showcase abilities that seem supernatural. Others will illuminate their fragile cellular machinery, their evolutionary ancestry, and their unexpected connections to our own existence. Throughout this exploration, I hope you'll discover a gentle, rhythmic sense of wonder that eases you toward rest. If you find value in what we share, please consider liking, subscribing, or leaving your reflections in the comments below. Your engagement helps us connect with others who share your curiosity. For now, though, settle in. Let your thoughts grow quiet and allow fascination to carry you gently toward sleep. Let us begin our journey into the microscopic marvels of the water bear. These creatures represent something profound, proof that life's boundaries stretch far beyond what we once imagined. In laboratories and natural environments alike, tardigrades defy our expectations at every turn. They challenge our understanding of what it means to be alive, what it means to endure, and what it means to persist across epochs that would erase nearly everything else. As we move through each fact tonight, picture yourself shrinking down to their scale, wandering through forests made of moss filaments, crossing oceans contained in dewdrops, and witnessing survival strategies so alien they seem borrowed from science fiction. Yet these are not fictional beings. They are real, abundant, and closer to you than you might think. They inhabit the gutters of your roof, the cracks in sidewalks, the soil of your garden. Wherever moisture can gather, however briefly, tardigrades find a way to thrive. Their story is both ancient and ongoing, a testament to the power of adaptation, patience, and the will to persist against all odds. Tardigrades possess the extraordinary ability to survive in outer space. During a 2007 experiment, researchers sent small populations of these microscopic animals into orbit, where they were exposed without any shielding to the vacuum surrounding Earth. No atmosphere, no oxygen, no warmth, just the unforgiving emptiness of space. Almost every other organism would die within moments under such conditions. The water bears, however, persisted. Remarkably, some not only survived the experience but also successfully reproduced after returning to Earth. This discovery astonished both biologists and space scientists, demonstrating that these tiny creatures can tolerate environments more hostile than almost anywhere on our planet. For tardigrades, the vacuum of space doesn't spell immediate destruction. Instead, it becomes merely another extreme condition they can endure in dormancy, waiting patiently for circumstances to improve. Their orbital survival establishes them as among the very few organisms capable of existing completely beyond Earth's protective atmospheric envelope. This raises intriguing questions about life's potential persistence in extraterrestrial environments. The mechanisms behind this space survival involve entering a state called the tun, which we'll explore more deeply later. In this form, their biological processes slow to near zero, and their bodies become resistant to radiation, vacuum, and temperature fluctuations. When conditions normalize, they rehydrate and resume activity as though awakening from ordinary sleep. The implications stretch our imagination. If these creatures can survive the harshness of space, might similar organisms exist elsewhere in the cosmos, drifting between worlds? Tardigrades also withstand temperatures far exceeding the boiling point of water. Most life forms perish when heated beyond a narrow thermal range. Their cellular machinery breaks down, proteins lose their structure, membranes rupture, and vital molecules disintegrate. Yet tardigrades have survived experimental exposures to water heated well beyond 100 degrees Celsius. Scientists remain uncertain about the complete mechanisms, but evidence suggests specialized protective proteins prevent critical structures from collapsing under thermal stress. This heat tolerance isn't absolute. Prolonged exposure kills many individuals. However, enough survive to demonstrate just how far their limits extend. The mental image of a creature smaller than a sand grain enduring temperatures comparable to volcanic environments transforms the tardigrade from a simple animal into something that feels paradoxical, almost impossible. Their survival at such extremes challenges our assumptions about the conditions necessary for life, suggesting that temperature ranges we consider lethal might merely be uncomfortable for properly adapted organisms. At the opposite thermal extreme, tardigrades demonstrate equally impressive cold tolerance. Laboratory experiments have exposed them to temperatures approaching absolute zero, the point where molecular motion virtually ceases. For most organisms, freezing proves fatal. Sharp ice crystals form within cells, puncturing membranes and destroying delicate internal architecture. Yet tardigrades in their dormant tun state resist this destruction with remarkable efficiency. Their bodies employ mechanisms that prevent catastrophic ice crystal formation. Internal chemistry appears to safeguard essential systems, keeping them intact even as temperatures plummet to levels never encountered in Earth's natural environments. When gradually thawed, these animals awaken slowly, as if emerging from an impossibly deep slumber. This ability to survive extreme cold suggests they could theoretically endure conditions on comets' icy surfaces or in the frigid depths of interplanetary space. Contemplating a microscopic creature surviving temperatures colder than any location naturally occurring on our planet expands our conception of habitability. What we consider life-friendly conditions may be far more flexible than we've traditionally assumed. The tardigrade's temperature tolerance spans from near absolute zero to above boiling, a range of several hundred degrees. This makes them among the most thermally resilient organisms ever studied. Perhaps their most celebrated capability involves complete dehydration. Most living things depend on water as life's fundamental requirement. It maintains cellular structure, enables biochemical reactions, and allows proper functioning of all vital processes. Without water, death typically arrives swiftly. Tardigrades, however, have evolved a remarkable solution. When their environment dries out, they enter suspended animation, losing up to 97% of their body water. In this desiccated state, specialized proteins and sometimes protective sugar molecules lock their cellular components in place, preserving them in a kind of biological stasis. They can remain this way for months, years, or even decades. Then, with just a single droplet of water, their tissues rehydrate and expand. They extend their stubby legs, activate their feeding apparatus, and continue living as though no time had elapsed. It appears they've discovered how to pause existence itself, effectively skipping over dangerous drought periods until favorable conditions return. This desiccation tolerance represents one of their most alien-seeming abilities. Imagine shutting down all biological processes, waiting in a state between life and death, then restarting completely when circumstances improve. For tardigrades, this isn't science fiction. It's evolutionary adaptation refined over hundreds of millions of years. The ton represents the tardigrade's ultimate survival mechanism. When environmental conditions become hostile, through drying, freezing, heating, or other stresses, the water bear retracts its 8 legs and compresses itself into a compact, barrel-shaped capsule. Inside this diminutive form, metabolic activity decreases to barely detectable levels, sometimes appearing to stop entirely. In the ton state, a tardigrade can withstand crushing pressures, intense radiation, and complete absence of oxygen. This transformation places the creature in a liminal zone between life and death, neither fully living nor truly deceased, but suspended in cryptobiotic animation. Scientists regard this as one of nature's most extreme examples of cryptobiosis, the ability to halt all life functions completely and then restart them when conditions improve. The ton is remarkably small and durable, capable of being transported on dust particles, soil grains, or fragments of moss, waiting silently for moisture to return. When environmental conditions become favorable again, the ton absorbs water and gradually unfurls. The tardigrade emerges from its protective state and resumes normal activity. This process can occur after hours, weeks, or even years in dormancy. The biological machinery required to achieve this feat involves complex molecular choreography that researchers are still working to fully understand. Special proteins stabilize cellular structures, preventing the damage that would normally occur during such dramatic metabolic shutdown. Some tardigrades have revived after remaining in the ton state for decades without any water. Consider falling asleep during a severe drought and awakening 30 years later when rain finally arrives. For these microscopic creatures, this isn't fantasy, it's documented reality. Scientists have recorded individuals that stayed in their dormant ton form for decades, then rehydrated and continued living as though nothing unusual had occurred. During this extended dormancy, their cells remain frozen in time. DNA stays protected through specialized mechanisms. Proteins remain stabilized in glass-like states, and all processes pause. The ability to endure decades without water astonishes researchers because most organisms can barely survive hours or days of complete dehydration. Yet tardigrades bridge entire human generations while in living stases, challenging our fundamental understanding of biological time. This capability raises fascinating questions that researchers continue to explore. What determines the maximum duration a tardigrade can remain dormant? Might there be undiscovered species within ancient moss specimens that have survived for centuries, waiting for the right conditions to revive? If such organisms exist, they would demonstrate that time, which governs nearly all living things so strictly, seems to bend and flex around the peculiar biology of water bears, operating by different rules entirely. Tardigrades resist radiation at levels that would prove instantly fatal to most life forms. Radiation remains invisible yet deadly, shattering DNA strands, breaking apart molecules, and disrupting the delicate chemistry that sustains life. Humans exposed to high radiation doses develop acute sickness and perish rapidly. Tardigrades, however, endure exposures hundreds of times greater than what humans can withstand, emerging largely unharmed. Laboratory testing has revealed they can survive ionizing radiation at levels that would sterilize most environments. The secret appears to lie in their extraordinary DNA repair mechanisms and unique proteins that shield genetic material from damage. Some researchers theorize that tardigrade cells can tolerate significantly more damage than our cells, providing time for repair systems to work before lethal breakdowns occur. This resilience has inspired research into whether tardigrade genes might inform better protective medicines for radiation exposure in humans. The image of such a delicate, gel-like creature weathering radiation storms capable of sterilizing entire landscapes provides perhaps the clearest evidence of their nearly impossible toughness. Their radiation resistance suggests they could survive nuclear disasters, cosmic ray bombardment during space travel, or exposure to radioactive materials that would devastate other organisms. This ability adds another layer to their reputation as nature's most resilient animals. Tardigrades also endure pressures exceeding those found in Earth's deepest ocean trenches. The Mariana Trench descends 11 kilometers beneath the Pacific Ocean surface, where pressures reach over 1,000 times those at sea level. Yet tardigrades have survived laboratory simulations of even greater crushing forces. Their ton state compresses their bodies into forms remarkably resistant to structural collapse. Cellular architecture in tardigrades appears uniquely stable under extreme pressure, avoiding the ruptures and failures that destroy most organisms. Imagine being subjected to forces powerful enough to crush a military submarine, yet surviving intact. Water bears, despite their microscopic size, handle such extremes routinely. Their pressure resistance suggests they could potentially survive in buried environments like deep rock pores, or possibly within the subsurface oceans that rise to exist on moons like Europa or Enceladus. While massive whales and sophisticated engineering struggle in the deepest ocean zones, these microscopic survivors remind us that life's greatest resilience often manifests in its smallest members. Size doesn't correlate with toughness. Sometimes the tiniest organisms possess the most remarkable survival capabilities, having evolved solutions to challenges that larger creatures cannot overcome. Tardigrades aren't merely survivors of harsh environmental conditions, they're survivors of geological time itself. Genetic analysis suggests their ancestors first emerged more than 500 million years ago during the Cambrian explosion, an era when life experimented with revolutionary new body plans and designs. While entire dynasties of organisms arose and disappeared, water bears persisted through it all. They existed as continents slowly drifted across the globe, as seas advanced and retreated repeatedly, as forests spread across barren landscapes, and as dinosaurs both emerged and vanished. Few organisms alive today can claim such an extraordinarily ancient lineage. When you observe a tardigrade under microscope magnification, you're not simply viewing a living creature, you're glimpsing a direct connection to Earth's most distant biological past. Their stubby legs may be microscopic, but they traverse a temporal scale that dwarfs all of human history. This deep time perspective reminds us that resilience can be measured not only in toughness against environmental extremes, but also in endurance across ages. The fact that tardigrades have remained relatively unchanged for hundreds of millions of years suggests they discovered an evolutionary strategy so successful that major modifications became unnecessary. Tardigrades have outlived all 5 major mass extinction events that punctuated Earth's history. These catastrophic episodes wiped out vast percentages of life on our planet. Dinosaurs vanished during one extinction event. Ancient trilobites disappeared during another. Yet tardigrades endured every single one of these global catastrophes. Whether caused by asteroid impacts, massive volcanic eruptions, dramatic climate shifts, or oxygen crises in ancient oceans, these tiny animals found ways to persist. Their ability to enter suspended animation likely helped them survive the worst environmental disasters in planetary history. Unlike many species requiring specific environmental conditions to thrive, tardigrades could simply wait out catastrophes in their dormant ton state. This means their lineage predates dinosaurs, predates flowering plants, and even predates many ocean basins in their current configurations. During every major ecological collapse Earth has experienced, tardigrades discovered survival strategies. This stubborn resilience suggests that if our planet were to suffer another catastrophic environmental shift, tardigrades would likely rank among the last organisms still persisting. Their track record across geological time provides compelling evidence that when conditions turn hostile, these microscopic survivors have already evolved the tools needed to endure, outlasting nearly everything else that walks, swims, or flies. Despite their widespread distribution and ancient lineage, tardigrades have left almost no fossil record behind. For organisms so abundant across the planet, their absence from geological strata seems paradoxical. Unlike shells, bones, or other hard structures that preserve well in rock, tardigrade bodies are soft, minuscule, and decompose rapidly after death. This means they vanish quickly from the geological record, leaving minimal traces for paleontologists to discover. The scarcity of tardigrade fossils makes reconstructing their evolutionary history exceptionally challenging. Scientists must rely primarily on living species and genetic analysis to piece together their ancient past. Only a handful of fossilized specimens have ever been confirmed, most preserved in amber, ancient tree resin that captured them as they crawled through moss millions of years ago. These rare amber inclusions provide precious windows into tardigrade evolution. Each newly discovered fossil feels like unearthing a lost chapter from an otherwise invisible story. The absence of evidence only deepens their mystery, making these tiny creatures seem like elusive phantoms that slipped almost unnoticed through Earth's geological ages. Without fossils to guide research, scientists must use sophisticated molecular techniques to estimate when different tardigrade lineages diverged and how they spread across the planet. The first confirmed tardigrade fossil wasn't discovered until 2021. For centuries, scientists knew tardigrades only from living specimens collected in contemporary environments. Then researchers announced the discovery of the first verified fossil species, Paradorophoribius chronocarabeus, trapped in Dominican amber approximately 16 million years old. This fossil preserved the water bear in stunning detail, revealing structures invisible in most preservation conditions. Researchers could examine its claws, mouth parts, and even subtle anatomical features that closely matched modern relatives. Finding such a specimen represented a monumental breakthrough because it provided a rare calibration point for understanding tardigrade evolutionary rates. The fossil demonstrated that even tens of millions of years ago, these animals already resembled their modern descendants remarkably closely. This discovery suggests that once tardigrades evolved their successful body plan, compact, adaptable, and extraordinarily resilient, very little needed to change over vast stretches of time. They became evolutionary survivors not only in function but also in form, maintaining their basic design across epochs that saw dramatic transformations in most other animal groups. That single drop of amber thus gave scientists their clearest glimpse yet of tardigrades frozen in deep geological time, confirming suspicions that their winning strategy has remained largely unchanged for millions of years. The earliest tardigrade ancestors are believed to have inhabited prehistoric oceans. Today we associate these creatures primarily with moss cushions and soil environments, but their origin story begins underwater. Early water bears likely crept along marine sediments, feeding on microbial mats in shallow, nutrient-rich waters filled with algae, bacteria, and primitive animal life. These ancient seas provided the evolutionary cradle where tardigrades first developed their distinctive characteristics. From these marine beginnings, tardigrades gradually expanded into brackish and freshwater habitats. Eventually, they colonized moist terrestrial environments, including moss patches and lichen colonies. This ancient oceanic ancestry explains many of their peculiar traits, particularly their continued reliance on thin water films to remain active. Even now, every land-dwelling tardigrade carries the legacy of its marine heritage, with biology still fundamentally tied to liquid water's presence. The journey from ancient seas to modern parking lot moss demonstrates the incredible evolutionary voyage these tiny animals have completed across habitats. Their transition to land represents one of the earliest animal colonizations of terrestrial environments, predating many larger and more familiar organisms by millions of years. Tardigrades were among the first animals to successfully colonize moist land environments. Long before vertebrates ventured onto dry ground, microscopic pioneers like tardigrades had already established themselves in damp terrestrial niches. Scientists believe they ranked among the earliest animals to exploit the delicate water films coating mosses, lichens, and soil particles in terrestrial settings. These habitats are inherently unstable. They dry and rehydrate frequently, shift with rainfall patterns, and experience dramatic temperature fluctuations. For tardigrades, however, this instability wasn't a barrier but rather a challenge they mastered through evolutionary innovation. Their ability to desiccate into protective tons made them perfectly suited for colonizing land environments where water could vanish suddenly and unpredictably. By successfully adapting to these conditions, tardigrades opened ecological doors to entirely new worlds, exploiting habitats inaccessible to less adaptable organisms. In many ways, their conquest of moist terrestrial environments foreshadowed later grander invasions of land by insects, amphibians, and reptiles. These tiny water bears essentially paved the way, demonstrating that life could thrive outside the ocean's protective embrace. They showed that with proper adaptations, the terrestrial realm offered vast opportunities for organisms willing to solve its unique challenges. Even with modern genetic tools, scientists continue debating exactly where tardigrades belong on the evolutionary tree of life. They clearly share relationships with arthropods, the group containing insects, spiders, and crustaceans, and with onychophrans, commonly known as velvet worms. However, the precise branching point remains disputed among researchers. Some analyses suggest closer kinship to insects, others hint at earlier evolutionary splits, and still others propose entirely unique placements. This uncertainty stems from tardigrades' unusual mixture of anatomical and genetic traits. They walk like miniature arthropods but possess simplified body structures that don't fit neatly into existing categories. Their genetic code contains unique elements not found in closely related groups. Additionally, their microscopic size and sparse fossil record complicate efforts to resolve their evolutionary position definitively. The debate continues as each newly sequenced tardigrade genome adds pieces to this complex puzzle. Until researchers reach consensus, tardigrades remain not only biological marvels but also evolutionary enigmas, their exact place in life's grand family portrait still drawn in tentative, uncertain outlines. Despite uncertainties, there's broad consensus that tardigrades share deep evolutionary roots with two fascinating groups, arthropods and velvet worms. All three likely descended from a common ancestor that lived hundreds of millions of years ago. This ancient kinship explains their jointed limbs, molting exoskeletons, and certain nervous system patterns that appear similar across these groups. Velvet worms are larger, slimy predators with stubby legs that hunt in tropical forests. Arthropods diversified into extraordinarily successful groups including crabs, beetles, and butterflies. Tardigrades remained tiny wanderers, but all three groups share fundamental biological similarities pointing to common ancestry. Understanding this connection adds surprising depth to tardigrade biology. When you observe their tiny claws under microscope magnification, you can imagine distant family resemblances to crab pincers or insect feet. This evolutionary kinship reminds us that tardigrades aren't isolated oddities but rather important members of a lineage that gave rise to some of Earth's most successful and diverse animals. In their microscopic bodies, tardigrades preserve echoes of the grand evolutionary saga that shaped life both on land and in the seas. They represent a branch that diverged early, maintained certain ancestral features, and developed unique adaptations that allowed them to persist across geological time, while their relatives experimented with different body plans and ecological strategies. A bold 2015 study suggested that tardigrades had acquired nearly 1/6 of their DNA from other species through horizontal gene transfer, a process where organisms incorporate genetic material from bacteria, fungi, or even plants. This claim was astonishing, implying that water bears had borrowed survival mechanisms from across the entire tree of life. However, subsequent research revised these numbers downward, suggesting the initial estimate was inflated. though confirming that at least some foreign genes are genuinely present. This ongoing debate highlights just how unusual tardigrade genomes truly are. Whether they borrow DNA directly from other organisms or independently evolved similar genetic solutions remains partly unresolved. Regardless of the mechanism, the result remains clear. Tardigrades possess genetic toolkits unlike any other animal group. If even portions of their resilience derive from this borrowed or convergent genetic material, they stand as living examples of evolution's remarkable inventiveness. They remind us that life sometimes shares its molecular secrets in unexpected ways, blurring the boundaries between species and creating genetic mosaics that challenge our traditional understanding of inheritance. This genetic flexibility may partly explain their extraordinary survival capabilities across such diverse extreme conditions. Among the strangest discoveries in tardigrade biology is the identification of proteins completely unlike those found in any other known organism. These unique proteins, such as DSUP, short for damage suppressor, appear to shield DNA from radiation and oxidative stress. Other specialized proteins help tardigrades enter their glassy, vitrified state during desiccation, protecting cellular structures when water disappears. These proteins are so unusual that scientists continue mapping out their molecular mechanisms at the most fundamental levels. Some have already been experimentally transplanted into other organisms, including cultured human cells in laboratory settings, where they conferred measurable protective benefits. This uniqueness suggests that tardigrades invented their own molecular survival equipment through evolution's long experiments in extreme living. The discovery also raises tantalizing possibilities for practical applications. If researchers can fully understand and perhaps harness these proteins, we might one day borrow strategies from the water bear's survival manual to protect ourselves in medicine, space exploration, or biotechnology. Imagine radiation therapies that cause less collateral damage, or preservation techniques that maintain cells in suspended animation for medical transplantation. The vast majority of tardigrades are almost impossibly small. Many species measure less than 0.5 millimeter in total length, meaning you could line up a dozen of them across the head of a standard pin. At this microscopic scale, they slip easily between individual soil grains, weave through moss fibers, and swim in dewdrops. To our unaided eyes, they remain completely invisible, their entire lives unfolding just beyond human perception's threshold. Yet under proper magnification, the blur of invisibility sharpens into a complete creature with legs, claws, a mouth, and even a distinctive lumbering gait. Their diminutive size reminds us that entire ecosystems thrive at scales we rarely consider or acknowledge. A hidden world exists where forests consist of moss stems and lakes are merely rainwater droplets. The tardigrade serves as a perfect ambassador to that realm. Tiny, easily overlooked, yet carrying resilience that outshines nearly everything larger. Not all of a tardigrade's defenses involve extraordinary superpowers. Sometimes simplicity works just as effectively. When startled or disturbed by rough handling, certain species curl themselves into tight, compact balls. They draw in their legs and tuck down their heads in a defensive posture. This behavior doesn't completely stop predators, but it makes the tardigrade harder to grasp and signals that the animal has entered a protective state. It represents a miniature version of what hedgehogs or pillbugs accomplish at larger scales. In the microscopic world, curling up can buy precious seconds, potentially confusing attackers and protecting softer tissues from immediate harm. This reminds us that despite their astonishing abilities, tardigrades also rely on simple, ordinary survival tricks, basic behaviors that mirror those of much larger animals across the tree of life. A tardigrade's movement represents one of its most charming features. With 4 pairs of stubby, jointed legs, they crawl and clamber in ways that feel oddly familiar, almost mammalian despite their microscopic dimensions. Under magnification, you can watch them lifting each foot in slow, deliberate steps as though carrying invisible weight. The coordination of 8 limbs gives them surprising control as they navigate moss forests and maneuver around soil particles, gripping surfaces with remarkable ease. Their gait is so recognizable and endearing that it directly inspired their nickname water bears. Scientists studying locomotion have even compared their walking patterns to those of insects many times larger, noting that similar coordination patterns appear in both groups. In this way, the tardigrade becomes a bridge between worlds, an animal so small it vanishes to the naked eye, yet moving with a rhythm we instinctively recognize as the careful, purposeful walk of a living being. Examining a tardigrade's leg under high magnification reveals delicate hooked claws curving outward from each foot. These claws serve as essential tools, allowing water bears to grip moss cells, anchor themselves to sediment particles, and pull their bodies forward with slow but steady progress. They're not weapons designed for tearing prey apart. Instead, they function as grappling hooks perfectly adapted for navigating a microscopic jungle. Different tardigrade species display variations in claw structure. Some possess long, slender claws while others have stubbier hooks. These subtle differences help taxonomists distinguish between closely related species. In the silent world of moss cushions and water films, those claws make the difference between drifting helplessly and climbing with intention and control. They may look simple under magnification, but they embody the essence of evolutionary adaptation, perfectly shaped for the miniature landscapes tardigrades call home. When Johann August Ephraim goes first described these creatures in 1773, he noticed something striking about their movement. Despite their tiny size, they walked like lumbering little bears. Their deliberate side-to-side steps, squat bodies, and sturdy legs evoked images of shaggy mammals wandering slowly through undergrowth. From this resemblance came the enduring nickname water bear, which has persisted for centuries. The name stuck perhaps because it captures both their appearance and their peculiar charm perfectly. Watching one march across a field of moss cells, you can almost imagine it as a miniature animal exploring a vast forest, completely unaware of its own microscopic scale. The name makes them instantly accessible and relatable, building a bridge between our world of large creatures and the hidden microcosmos where tardigrades actually live. It reminds us that even at microscopic scales, movement can feel familiar, endearing, and oddly relatable to our own experiences. In addition to water bear, tardigrades carry another popular nickname, moss piglets. This name emphasizes their plump, rounded bodies and the fact that they're frequently found crawling among moss cushions. With their squat proportions and snuffling mouths, they do somewhat resemble tiny piglets exploring a green barnyard composed of moss leaves. The term adds a playful twist to their image, contrasting their incredible toughness with a name that sounds almost comical. Moss piglet has become particularly popular in educational settings and children's books, helping young learners imagine these microscopic creatures as friendly, approachable beings rather than intimidating scientific specimens. For researchers, the name is less formal but no less affectionate. It demonstrates how even the most extraordinary survivors in biology can carry an image that's humble, even cute. These tiny moss piglets may be tougher than trained astronauts and able to survive conditions that would destroy most life on Earth, but they remain deeply endearing in their small, chubby form, a combination of toughness and charm that makes them favorites among scientists and educators alike. Under microscope magnification, most tardigrades reveal their inner workings without requiring dissection or chemical staining. Their bodies are naturally transparent, allowing scientists and curious students to observe organs and internal movements in real time. You can watch their digestive tracts fill with green algae as they feed, observe eggs forming inside females, and even see the pumping motion of their pharyngeal bulb as it draws in food. Transparency isn't unique to tardigrades. Many small organisms share this characteristic. However, in water bears it adds significantly to their fascination because it makes their survival mechanisms visible to observers. Watching a ton shrink into its compact form or an embryo slowly developing within an egg feels like observing nature's most delicate secrets without any interruption or obstruction. Their see-through bodies transform the microscope into a window rather than just a magnifying tool, giving us direct access to biological processes usually hidden inside opaque skin and bone. This transparency makes tardigrades exceptionally valuable for educational purposes and scientific research, as their internal biology becomes a living demonstration visible to anyone with basic equipment. In certain conditions, a tardigrade's digestive system can emit a faint glow when viewed under fluorescent microscopes. This luminescence often originates from the food they've consumed, such as algae or bacteria, which may naturally contain pigments that respond to specific light wavelengths. The result creates a shimmering, almost otherworldly view of a creature illuminated from within by its own gut contents. This isn't natural bioluminescence in the traditional sense, but rather an artifact of how certain substances interact with scientific equipment and specific lighting conditions. Still, the effect proves mesmerizing. A transparent body with glowing channels resembling a tiny lantern drifting across a microscope slide. These glowing guts remind us that even the smallest animals can create moments of unexpected beauty, especially when modern tools allow us to observe them in ways nature never intended. For students and researchers alike, this phenomenon adds a spark of visual wonder to the already astonishing story of the water bear. It transforms routine observation into something magical, helping maintain engagement and curiosity during extended study sessions. Tardigrades are not blind wanderers navigating their environment randomly. Many species possess a pair of eye spots near the front of their bodies. These aren't complex visual organs like human eyes, but simple light-detecting structures capable of distinguishing between light and darkness. They help the animals orient themselves, avoiding intense illumination that might dry them out while seeking the safety of shaded, moist environments. The eye spots consist of little more than clusters of pigmented cells, yet they serve crucial survival roles. To observers, They appear as tiny black dots just visible under magnification. It's fascinating to consider that a creature so small carries a sensory system, primitive yet effective, to navigate its miniature world using light as a guide. Their eye spots hint at the universality of light as a navigational guide for life, whether in forests, oceans, or water films coating moss surfaces. Even at microscopic scales, vision, however simple and basic, shapes existence and influences behavior in measurable ways. Not every tardigrade species relies on light detection for navigation. In species inhabiting deep soil layers or sediments where light never penetrates, eye spots would serve no purpose and have been lost through evolution. These species are completely eyeless, navigating instead through touch and chemical cues detected by other sensory structures. Without light to guide them, they rely entirely on their claws and legs to feel their way forward, exploring environments step by step through physical contact. Their lack of eyes demonstrates tardigrade adaptability. Evolution shapes each species for its particular ecological niche, adding or removing traits as environmental conditions demand. In the darkness of soil grains or murky depths of sediments, eyes offer no selective advantage. Therefore, tardigrades in these habitats thrive perfectly well without them. This reminds us that what seems essential to humans, like vision, is not universal across all life. For water bears, survival comes not necessarily from seeing the world, but from enduring it through whatever sensory mechanisms prove most useful in their specific environment. Unlike many animals, tardigrades don't possess blood coursing through vessels and veins. Their bodies are so small and structurally simple that they don't require circulatory systems. Instead, nutrients and gases diffuse naturally through their tissues, carried in the water surrounding and permeating them. This absence of blood makes them seem almost alien, as if stripped down to the most minimalist design for sustaining life. For scientists, it also makes them easier to study under microscopes because nothing opaque obscures their internal organs. The lack of blood hasn't hindered their evolutionary success whatsoever. Instead, it demonstrates how many different solutions evolution discovers for the same fundamental problem of sustaining life. In a way, tardigrades remind us that what we consider universal features, blood pumping through a heart, for instance, are not actually requirements for survival. These water bears walk their ancient evolutionary paths without ever needing the crimson currents we take for granted in our own bodies. Instead of blood, tardigrades rely on a simple body cavity filled with circulating fluid. This liquid bathes their organs directly, carrying nutrients, metabolic waste products, and dissolved gases across tiny distances. In scientific terminology, this type of system is called a hemocoel, an open cavity rather than a closed network of veins and arteries. For tardigrades, this simple arrangement provides everything they need. Their minuscule size ensures that diffusion across the fluid happens quickly enough to sustain all life processes efficiently. Under microscope magnification, this internal environment appears as a shimmering, transparent sea inside the animal, a microcosm within a microcosm. It's a vivid reminder that complex circulatory systems represent luxuries for larger creatures, not fundamental requirements for all life. Water bears thrive with this ancient, minimalist design, providing proof that life can flourish with the simplest of internal plumbing as long as the organism's scale remains small enough and the biological strategy remains sound. When we think of animals, we often picture specialized organs, lungs rhythmically inflating, hearts beating steadily, but tardigrades manage without either structure. Their tiny bodies eliminate the need for specialized pumps or dedicated airways. Instead, oxygen diffuses directly through their surface membranes and into their cells, while nutrients drift freely in their body fluids without requiring active circulation. With no lungs to fill and no heart to beat, their internal rhythm is silent and passive. This simplicity is one reason they can shrink into a ton so effectively. With no elaborate organs to protect during desiccation, their body becomes easier to preserve during environmental stress. It's almost as though they represent a minimal blueprint for animal life: legs, a nervous system, a digestive tract, but none of the extra machinery that larger animals require. And still, despite lacking these essential organs, they endure conditions that defeat far larger, more complex organisms. The absence of lungs or heart is not a weakness or evolutionary deficiency, but rather part of the elegant simplicity that makes their extreme resilience possible in the first place. In tardigrades, breathing doesn't involve inhaling or exhaling in any conventional sense. Oxygen seeps gently through their cuticle, the flexible outer covering serving as their skin. This diffusion process works efficiently because they are so small. Oxygen molecules don't have far to travel before reaching vital cells requiring respiration. Their cuticle functions not just as a protective barrier, but also as a gateway, allowing gases to move in and out freely based on concentration gradients. This method ties them closely to water and moisture. Without a film of liquid coating their bodies, diffusion fails, which is precisely why they must rely on their ton state during drought periods. Watching them through a microscope, it's strange to realize that no breath stirs within them. No air passes visibly in or out. Yet life continues silently, with oxygen molecules slipping invisibly across a thin membrane. This quiet way of breathing illustrates how different yet equally effective survival strategies can be when life adapts to microscopic scales where surface area to volume ratios make passive diffusion practical. Despite their overall simplicity, tardigrades possess a brain, a small cluster of nerve cells situated above the esophagus. What fascinates scientists is that its structure resembles that of arthropods, their distant evolutionary cousins. Nerve cords extend down the length of their bodies, connecting to ganglia in each body segment, a layout that feels like a miniature version of an insect's nervous system. This structural resemblance strengthens the evolutionary link between tardigrades and larger jointed creatures, providing anatomical evidence to complement genetic data. Under microscope magnification, the brain itself isn't grand or elaborate, but its function remains essential for coordinating movement, processing sensory input, and controlling feeding behavior. For an animal so microscopically small, the presence of such neurological organization is remarkable and somewhat surprising. It suggests that even at the tiniest scales, nervous systems can reflect ancient organizational patterns echoing through evolutionary time across diverse animal groups. In the water bear's brain, we glimpse not just a control center for basic functions, but a living map of its deep kinship to other creatures. The neural architecture carries evolutionary signatures linking these microscopic survivors to the vast diversity of arthropods that dominate terrestrial and aquatic ecosystems worldwide. Tardigrade reproduction produces some of the most beautiful microscopic objects in nature: their eggs. Female water bears lay eggs either directly into their surrounding environment or within the shed cuticle remaining from their molting process. Under magnification, these eggs often display intricate ornamentation and surface patterns. Some appear smooth and relatively simple, but many exhibit elaborate ridges, bumps, spines, or delicate sculpting across their surfaces. These patterns aren't merely aesthetically lovely. They also serve practical purposes for scientists who use egg morphology to identify and distinguish between species. Each egg becomes a tiny masterpiece, hinting at both functional advantages and unintentional artistry. In educational settings, students are often astonished to discover how much structural variety exists in such minuscule spheres of life. The ornate shells protect developing embryos inside, giving the next generation enhanced chances to endure environmental challenges from the moment of formation. Even at scales measured in fractions of millimeters, life finds ways to combine structural strength, practical functionality, and unexpected beauty into its reproductive designs. Among the many varieties of tardigrade eggs, some species produce truly spectacular structures. These eggs are covered with long, sharp spines or branching filaments, making them look like alien spheres bristling with defensive projections. The spines likely serve multiple purposes: discouraging predators from consuming them, anchoring eggs securely to surfaces, or helping them remain stationary in water currents until hatching occurs. Under microscope magnification, they resemble miniature sea urchins or fantastical seed pods from science fiction narratives. Scientists marvel at the diversity of egg morphologies, noting how each species develops its own signature egg style, ranging from smooth and featureless to extravagantly spiked with complex ornamentation. For observers, this adds another layer of wonder to the already improbable story of tardigrades. Not only are adult water bears nearly indestructible, but even their eggs carry visual uniqueness that feels otherworldly and alien. It's as if their resilience begins before life itself has properly commenced, with protective structures surrounding the next generation from its earliest developmental stages. In certain tardigrade species, females protect their eggs by laying them inside the old cuticle they shed during molting cycles. This discarded skin becomes a protective shelter, keeping eggs safe from predators, desiccation, and other environmental hazards. The strategy is both simple and remarkably effective, recycling a cast-off body part into a defensive capsule for offspring. Under microscope observation, you can sometimes see clusters of eggs nestled inside the translucent husk of a parent shed skin, appearing like pearls carefully arranged in a fragile case. When young tardigrades hatch, they emerge into the world already having been shielded during their most vulnerable developmental stage. This behavior highlights the quiet ingenuity of tardigrades, using what they already possess to safeguard future generations without requiring additional energy expenditure or specialized structures. Even at this tiny scale, parental care finds expression, ensuring that life continues against considerable odds in unstable microhabitats. When tardigrade eggs finally hatch, the young don't emerge as larvae or worm-like infants requiring metamorphosis. Instead, They are miniature versions of adults, complete with legs, claws, and the immediate ability to move and feed independently. These hatchlings measure just fractions of a millimeter, but they already carry the full anatomical blueprint of the water bear body plan. Over time, they grow by molting, shedding their exoskeletons periodically to expand in size. However, from the moment they emerge from eggs, they are unmistakably tardigrades with all essential structures already functional. Watching them under microscope magnification feels like witnessing a secret miracle. Tiny creatures awakening inside fragile shells, ready to walk in the microcosmos immediately upon hatching. This precocial development means they can survive independently from birth without extended parental care or protection. It represents another survival strategy. Each new tardigrade steps into the world fully prepared for the environmental challenges it will immediately face, rather than requiring a vulnerable larval stage that might not survive harsh conditions. In certain tardigrade populations, females far outnumber males, sometimes to the point where males are rarely observed at all. This skewed sex ratio may link to their reproductive strategies. Some species are capable of parthenogenesis, reproducing without males through asexual means, while others simply maintain female-dominated populations to sustain overall numbers more efficiently. For scientists, these ratios provide clues to how populations adapt in environments where finding mates proves difficult due to low population density or spatial isolation. For the tardigrades themselves, it represents another example of reproductive flexibility. If environmental circumstances make one strategy more efficient than another, evolution provides alternative pathways. The prevalence of females highlights how different survival and reproduction can appear in the microscopic world. While many larger animals depend on balanced populations of both sexes for reproductive success, tardigrades demonstrate that life finds alternate reproductive paths, reshaping even fundamental biological processes like sexual reproduction to fit the harsh realities of their tiny, unstable ecosystems. High in mountains where moss clings precariously to rocks and the atmosphere thins considerably, tardigrades live in delicate films of water coating each leaf and stem. These environments are harsh and unforgiving, exposed to wind, frigid nights, and blazing sun during daylight hours. Yet water bears thrive there despite these challenges. The thin water layers can vanish quickly under intense sun or dry wind, but tardigrades respond by entering their tun state, enduring desiccation until clouds roll in or rain returns moisture. In this way, mountaintop moss cushions become miniature kingdoms filled with some of Earth's toughest microscopic residents. Imagining them crawling across single moss cells at altitudes where trees no longer grow brings valuable perspective. Resilience isn't always about strength measured in size, but about patience expressed in smallness. In places where only the hardiest plants can survive, these tiny animals persist as quietly as they always have, turning mountaintops into silent strongholds of microscopic life that most visitors never notice. At the opposite extreme, far beneath ocean waves, tardigrades have been discovered near hydrothermal vents, places where superheated water bursts from cracks in the ocean floor. Pressures here are immense, darkness is absolute, and temperatures swing wildly between scalding vent water and near freezing surrounding seawater. Yet tardigrades inhabit even these extreme environments. They live in sediments near these geological features, demonstrating that their distribution spans from the highest terrestrial elevations to the deepest ocean zones. This range makes them among the most widely distributed animal groups on Earth. Whether in tropical rainforests, Arctic tundra, mountain peaks, or abyssal depths, tardigrades have found ways to colonize nearly every conceivable habitat where even trace amounts of water exist periodically. Their presence in such diverse environments underscores their remarkable adaptability. They don't just survive extremes in laboratory conditions, they actually inhabit these places naturally, thriving in niches that remain inaccessible or lethal to most other organisms. Tardigrades don't limit themselves to pristine wilderness environments. They also thrive in human-altered urban landscapes. You can find them in roof gutters, sidewalk cracks, parking lot moss patches, and even window box gardens. Wherever moisture can briefly accumulate, in morning dew, after rainfall, or in humid microclimates, tardigrades establish populations. This urban distribution means they live closer to humans than most people realize. The moss growing on your fence, the lichen on park benches, the damp soil in planters, all potentially harbor thriving tardigrade communities. Their ability to colonize disturbed habitats demonstrates impressive ecological flexibility beyond their physiological resilience. For educators, this proximity makes tardigrades excellent subjects for citizen science projects. Students can collect samples from their own neighborhoods, discovering that remarkable microscopic life exists not just in exotic locations, but literally in their own backyards. This accessibility helps build connections between abstract scientific concepts and tangible, observable reality. Tardigrades inhabit every continent on Earth, including Antarctica. On the frozen southern continent, they live in ice-free areas where mosses and lichens manage to grow during brief summer periods. Despite temperatures that plummet far below freezing for most of the year, tardigrade populations persist in these marginal habitats, entering ton states during winter months and reactivating when conditions temporarily improve. Their Antarctic presence demonstrates that geographic barriers mean little to organisms capable of entering cryptobiosis. They've colonized Earth's most isolated landmasses, likely transported by wind, migrating birds, or other natural dispersal mechanisms. This global distribution makes them among the most cosmopolitan animal groups known to science. Researchers studying biogeography find tardigrades valuable because their wide distribution patterns help reveal how microscopic organisms spread across the planet. Unlike larger animals limited by obvious physical barriers, tardigrades can potentially travel vast distances in dormant states, waiting to reactivate wherever they happen to land if conditions prove suitable. Some tardigrade species have adapted specifically to freshwater environments. They inhabit ponds, lakes, streams, and rivers, crawling along submerged vegetation and sediments. These aquatic species differ somewhat from their terrestrial relatives, having evolved specialized adaptations for permanently wet environments rather than intermittent moisture. Freshwater tardigrades still possess the ability to enter ton states, but they may rely on it less frequently than terrestrial species do. Their environments can still become hostile through pollution, temperature extremes, or oxygen depletion, triggering cryptobiosis as a survival response. Studying these aquatic species helps researchers understand how tardigrade biology varies across different habitat types. Lakes and ponds around the world harbor diverse tardigrade communities, often with multiple species coexisting in the same water body. Each species occupies slightly different ecological niches, some preferring sandy bottoms, others favoring algae mats or aquatic moss. This diversity adds complexity to freshwater ecosystems that often goes unnoticed without microscopic investigation. Other tardigrade species remain exclusively marine, living in ocean sediments and on underwater surfaces. These marine tardigrades represent the ancestral lifestyle, maintaining the ecological roles their ancient forebears occupied hundreds of millions of years ago. They crawl through sand grains, cling to algae, and navigate the spaces between sediment particles in shallow and deep waters. Marine species display remarkable diversity in body forms and ecological strategies. Some are predators hunting smaller organisms, while others graze on bacterial films or consume detritus. Their presence in ocean ecosystems contributes to nutrient cycling and energy flow, though their small size means their ecological impact often gets overlooked in broader marine research. Studying marine tardigrades provides insights into the group's evolutionary origins and helps scientists understand how terrestrial species evolved from marine ancestors. The transition from ocean to land represents one of evolution's major innovations, and tardigrades offer a living example of organisms that successfully made this journey while maintaining populations in both realms. Tardigrades have been found living in soil samples from deserts where rain might not fall for years. These desert-dwelling populations spend the vast majority of their existence in tund states, waiting through extended droughts that would eliminate most other organisms. When rare rains finally arrive, they rehydrate rapidly, feed, reproduce, and potentially enter dormancy again before moisture disappears. This feast or famine lifestyle requires extreme patience on biological timescales. Desert tardigrades may experience only brief windows of active life scattered across decades of dormancy. Yet their populations persist, demonstrating that active metabolism isn't always necessary for long-term species survival. Sometimes waiting proves more effective than constant activity. Their desert presence also raises questions about the upper limits of dormancy duration. If tardigrades can survive years without water in desert soils, might some survive even longer under optimal preservation conditions? Researchers continue investigating these limits, searching for the boundaries of cryptobiotic endurance. In tropical rainforests, tardigrades thrive in the constantly moist environments provided by canopy mosses, leaf litter, and humid soil layers. Unlike their desert relatives, rainforest tardigrades may rarely need to enter tund states because moisture remains relatively constant. This allows for more continuous life cycles with regular feeding, growth, and reproduction. Rainforest environments harbor exceptional tardigrade diversity, with numerous species coexisting in the same forest. Each species occupies distinct microhabitats, some preferring bark surfaces, others living in suspended mosses, still others inhabiting forest floor leaf litter. This niche partitioning allows multiple species to coexist without directly competing for identical resources. Studying tropical tardigrade communities helps researchers understand how these organisms diversify and adapt to stable versus variable environments. The contrast between rainforest and desert species illustrates evolution's flexibility in shaping organisms for radically different ecological challenges using the same basic body plan. Tardigrades even inhabit temporary water bodies like puddles, bird baths, and water-filled tree hollows. These ephemeral habitats can vanish within hours or days as water evaporates, yet tardigrade populations persist through repeated cycles of flooding and drying. Their ton capabilities make them ideally suited for such unpredictable environments. In these temporary habitats, tardigrades must complete their life cycles rapidly during wet periods, reproducing before water disappears. This creates selection pressure for fast development and efficient reproduction. Species inhabiting ephemeral waters often show adaptations for quick maturation, allowing them to capitalize on brief favorable conditions. These temporary habitats also serve as stepping stones for tardigrade dispersal. As puddles dry completely, dormant tardigrades may be transported by wind or animals to new locations. When they eventually encounter moisture again, perhaps in a completely different geographic area, they reactivate and establish new populations, contributing to their global distribution. Some tardigrade species demonstrate impressive climbing abilities, ascending vertical surfaces using their clawed feet. On moss-covered trees or rock faces, they navigate upward against gravity, gripping tightly to prevent falling. This climbing behavior allows them to exploit three-dimensional habitats more fully, accessing food sources and microhabitats unavailable to less mobile organisms. Their claws function like miniature grappling hooks, engaging with microscopic surface irregularities invisible to human eyes. The coordination required to climb while maintaining 8 points of contact demonstrates their neurological sophistication despite their simple brain structure. Watching them ascend moss stems under magnification reveals purposeful, controlled movement rather than random wandering. This mobility contributes to their ecological success. Rather than passively waiting for favorable conditions where they happen to be, tardigrades can actively seek out better microhabitats within their immediate environment, moving toward moisture, food, or shelter as needed. Tardigrades occupy an important ecological role as consumers of bacteria, algae, and plant cells. By feeding on these organisms, they help regulate microbial populations and contribute to nutrient cycling in soil and aquatic ecosystems. Their feeding activities break down organic matter, making nutrients available for other organisms in the food web. In moss and lichen communities, tardigrades help maintain ecosystem balance. Without predators to control bacterial and algal populations, these microorganisms might overgrow and alter habitat conditions. Tardigrades provide natural population control, creating more stable community dynamics in their microscopic ecosystems. Their ecological importance often goes unrecognized because of their small size and hidden lifestyles. However, in terms of biomass and metabolic activity, tardigrade populations can be substantial in favorable habitats. Their collective impact on ecosystem processes may be more significant than their individual size suggests. Some tardigrade species are carnivorous, preying on smaller organisms including nematodes, rotifers, and even other tardigrades. These predatory species possess stronger, more pointed stylets in their mouths, allowing them to pierce prey and extract body fluids. Their hunting behavior involves active pursuit rather than passive grazing. Carnivorous tardigrades demonstrate that even at microscopic scales, predator-prey dynamics shape community structure. They occupy higher positions in microbial food webs, controlling populations of smaller organisms just as larger predators do in more visible ecosystems. This adds complexity to our understanding of their ecological roles. Observing predatory behavior under microscopes reveals surprisingly sophisticated hunting strategies. Some species appear to stalk prey, approaching slowly before striking. Others may ambush prey from concealed positions. These behaviors suggest greater behavioral complexity than might be expected from such simple nervous systems. Cannibalism has been documented in some tardigrade populations under stressful conditions. When food becomes scarce or populations grow too dense, individuals may consume eggs or even other adults of their own species. This behavior, while seemingly brutal, may serve important survival functions. Cannibalism reduces competition for limited resources, potentially allowing some individuals to survive when food sources become insufficient for entire populations. From an evolutionary perspective, sacrificing some individuals may preserve genetic lineages that would otherwise disappear entirely during extreme resource scarcity. In laboratory cultures, cannibalism sometimes appears when researchers maintain populations at unnaturally high densities. This suggests it represents a stress response rather than normal behavior under natural conditions. Nonetheless, the capacity for cannibalism reveals flexibility in feeding strategies that may contribute to survival during exceptional circumstances. Despite their impressive defenses, tardigrades do face predation from various organisms. Nematodes, larger amoebas, and certain other microscopic predators consume tardigrades when they encounter them. The tardigrade's ability to curl into a defensive ball or enter a ton state may deter some predators, but these defenses aren't universally effective. In aquatic environments, small crustaceans and insect larvae occasionally consume tardigrades. Their soft bodies provide nutritious food for organisms capable of catching and consuming them. This predation pressure may influence tardigrade behavior, encouraging them to remain concealed in moss or sediment rather than wandering openly. The existence of natural predators reminds us that despite their extraordinary resilience to environmental extremes, tardigrades remain vulnerable to biological threats. Their superpowers protect them from physical and chemical stresses, but they haven't evolved complete immunity to being eaten. This places them firmly within normal ecological food webs despite their unusual abilities. Fungi pose another threat to tardigrade populations. Certain fungal species parasitize tardigrades, infecting them and eventually killing them. Fungal spores may attach to tardigrade cuticles, germinate, and penetrate their bodies. Once inside, the fungus consumes the tardigrade from within, eventually producing new spores that spread to infect other individuals. These parasitic relationships demonstrate that tardigrades, for all their resilience, still face biological enemies capable of exploiting vulnerabilities. Fungal infections may regulate tardigrade population densities in some environments, preventing unchecked population growth. This interaction adds another layer to the complex ecological relationships in microscopic communities. Researchers studying these parasitic fungi gain insights into tardigrade immunology and resistance mechanisms. Understanding how some individuals resist infection while others succumb could reveal additional aspects of their remarkable biology. Tardigrades were first discovered in 1773 by German zoologist Johann August Ephraim Goes. While examining samples of moss water under early microscopes, he observed these tiny creatures walking with their characteristic bear-like gait. Fascinated by their appearance and movement, he called them little water bears, a name that persists in various forms to this day. Go's discovery opened scientific awareness to an entire realm of microscopic animal life previously unknown. His descriptions, though lacking modern precision, captured the essential character of these organisms: their stubby legs, deliberate movement, and preference for aquatic moss habitats. This initial discovery sparked curiosity among naturalists of the era. Though tardigrades would remain relatively obscure for many decades, early microscopes lacked the resolution to reveal their finer anatomical details, limiting how much researchers could learn. Nonetheless, Go's observations established tardigrades as legitimate subjects of scientific inquiry. The scientific name tardigrada was coined in 1777 by Italian biologist Lazzaro Spallanzani. The name derives from Latin words meaning slow walker, referring to their deliberate, unhurried gait. Spallanzani conducted some of the earliest experiments on tardigrade resilience, subjecting them to desiccation and observing their revival upon rehydration. His work represented pioneering research into cryptobiosis, though the term wouldn't be invented for nearly 2 more centuries. Spallanzani recognized that these creatures could somehow suspend life processes and later restart them, a phenomenon that defied contemporary understanding of biology. His experiments laid groundwork for future investigations into their extraordinary survival mechanisms. The name tardigrada has endured as the formal designation for this phylum, encompassing over 1,300 described species. It captures an essential characteristic: their slow, careful movement, that remains one of their most recognizable features under microscopic observation. For over a century after their discovery, tardigrades remained curiosities studied by relatively few specialists. Taxonomy developed slowly as researchers identified new species and attempted to classify them based on limited anatomical features visible under available microscopes. Much early work focused simply on documenting their distribution and basic biology. The advent of electron microscopy in the mid-20th century revolutionized tardigrade research. Suddenly, scientists could observe ultrastructural details impossible to see with light microscopes. This revealed previously unknown anatomical features, allowing more sophisticated taxonomic classifications and deeper understanding of their unique adaptations. Modern molecular techniques have further transformed the field. DNA sequencing allows researchers to investigate evolutionary relationships, identify cryptic species that look identical but are genetically distinct, and uncover the genetic basis of their survival mechanisms. Tardigrade research has accelerated dramatically in recent decades as new tools reveal their secrets. Today, tardigrades enjoy popularity extending beyond scientific circles. They've become cultural icons representing resilience and survival against impossible odds. Their image appears on merchandise, in popular science media, and in educational materials worldwide. This popularity helps engage public interest in science while highlighting genuinely remarkable biology. Their cute appearance, the plump body, stubby legs, and bear-like waddle, makes them accessible and appealing despite being microscopic. Unlike many scientific subjects that seem distant or abstract, tardigrades feel relatable and endearing. This combination of toughness and charm makes them perfect ambassadors for microscopic life. Scientists welcome this public interest because it creates opportunities for outreach and education. When people feel curious about tardigrades, they become more receptive to learning about evolution, adaptation, extremophiles, and microscopic ecosystems, concepts that might otherwise seem dry or intimidating. Over 1,300 tardigrade species have been formally described and named by scientists, but researchers estimate the true number may exceed 10,000 species worldwide. Many habitats remain poorly sampled, particularly in tropical regions, deep ocean sediments, and remote areas. Each new survey typically reveals previously unknown species, suggesting vast undiscovered diversity. This hidden diversity exists partly because tardigrades are difficult to study. Their small size requires specialized equipment. Their taxonomy relies on subtle morphological details, and species identification often requires expert knowledge. Many potential tardigrade researchers lack access to necessary microscopy equipment or taxonomic expertise, leaving much biodiversity undocumented. As molecular techniques become more accessible, species discovery rates are accelerating. DNA barcoding allows researchers to identify distinct genetic lineages that may represent separate species despite looking nearly identical. This reveals cryptic diversity invisible to traditional morphological approaches, suggesting the true species count may be even higher than current estimates. Tardigrades are divided into three main classes based on anatomical features: Eutardigrada, Heterotardigrada, and Mesotardigrada. Eutardigrada represents the largest group, containing species with simple cuticles lacking elaborate ornamentation. Many freshwater and terrestrial species belong to this class, including some of the most commonly encountered and studied species. Heterotardigrada includes species with more complex cuticles, often featuring plates, spines, or other ornamentation. This class contains many marine species, though terrestrial representatives exist as well. Their elaborate cuticle structures may provide protection from predators or environmental stresses, though exact functions remain debated. Mesotardigrada contains only a single known species, discovered in a hot spring in Japan. This enigmatic class shares features with both other groups while possessing unique characteristics. Its isolated position suggests it may represent an ancient lineage that diverged early in tardigrade evolution, though its exact relationships remain uncertain. The smallest known tardigrades measure less than 100 micrometers in length, barely larger than a single human cell. These diminutive species push the lower limits of animal body size, approaching physical constraints imposed by cellular and molecular machinery. At such small scales, maintaining distinct organs and complex structures becomes increasingly challenging. Despite their tiny size, these miniature tardigrades retain all essential body systems: nervous system, digestive tract, reproductive organs, and musculature. This demonstrates remarkable efficiency in biological organization, packing necessary functions into extraordinarily compact forms. Studying these smaller species helps researchers understand minimum requirements for animal body plans. Their minute dimensions also create unique ecological opportunities. They can inhabit spaces unavailable to larger tardigrades, exploiting microhabitats between sand grains or within soil pore spaces too small for competitors. This niche partitioning allows multiple species to coexist in the same general environment without direct competition. The largest tardigrades can reach lengths exceeding 1.5 millimeters, massive by tardigrade standards, though still barely visible to unaided human eyes. These giants of the tardigrade world are big enough to see as tiny moving specks under good lighting conditions, though identifying them still requires magnification. Their larger size provides certain advantages, including greater physical strength and larger eggs producing more robust offspring. Larger species may have different ecological roles compared to their smaller relatives. They can consume bigger food particles, potentially prey on larger organisms, and may be less vulnerable to certain predators. However, their size also creates challenges. They require more food to sustain metabolism, face greater desiccation challenges due to lower surface area to volume ratios, and may be more visible to predators. The size range spanning from 100 micrometers to over 1,500 micrometers represents more than tenfold variation within a single animal group. This diversity in body size allows tardigrades to exploit different ecological niches while maintaining their fundamental body plan and survival strategies. Most tardigrade species are herbivorous or detritivorous, feeding primarily on plant cells, algae, bacteria, and decaying organic matter. They use their specialized mouthparts, a buccal tube containing sharp stylets, to pierce cell walls and extract contents. This feeding mechanism works like a microscopic syringe, puncturing targets and sucking out nutrients. Herbivorous species often show preferences for particular food sources. Some specialize on specific algae species. Others prefer moss cells, and still others consume bacterial films coating surfaces. These dietary preferences may reduce competition between species sharing the same habitat, allowing multiple tardigrades to coexist by partitioning available food resources. Their feeding activities contribute to nutrient cycling in microscopic ecosystems. By breaking down plant cells and processing organic matter, tardigrades help release nutrients that become available for bacterial decomposition and eventual recycling into new plant growth. This makes them important functional components of soil and aquatic ecosystems despite their small size. The tardigrade mouth contains a muscular pharynx that operates like a pump, creating suction to draw in food. This pharyngeal bulb is one of the most distinctive anatomical features visible under microscopes, often appearing as a pulsating structure when tardigrades feed actively. The pumping motion is rhythmic and mesmerizing to observe. Inside the mouth, sharp stylets made of hardened cuticle material pierce food sources. These stylets can penetrate tough algal cell walls or even pierce small animal prey in carnivorous species. Their effectiveness depends on both their physical sharpness and the muscular force applied by the pharyngeal apparatus. After feeding, tardigrades digest food in a simple tubular gut that runs the length of their body. Digestion occurs relatively quickly, with waste products expelled through a posterior opening. The efficiency of their digestive system allows them to extract maximum nutrition from limited food sources. important for organisms living in resource-poor microscopic habitats. Some tardigrade species can survive complete starvation for months while remaining active. Unlike entering cryptobiosis, these individuals maintain normal metabolism and movement despite lack of food intake. They appear to metabolize internal energy reserves extremely efficiently, prolonging survival during temporary food shortages. This starvation resistance provides advantages in unpredictable environments where food availability fluctuates. Rather than immediately entering dormancy when food becomes scarce, tardigrades can continue searching for new food sources while slowly depleting reserves. Only when starvation becomes prolonged do they eventually enter tun states. The metabolic mechanisms allowing such extended starvation remain incompletely understood. Researchers suspect they involve highly efficient energy metabolism, ability to catabolize and recycle cellular components, and perhaps reduced metabolic rates during food scarcity even while remaining active. Understanding these mechanisms could have applications for long-term food preservation or metabolic therapies. Tardigrades must molt periodically to grow, shedding their cuticles as they increase in size. This molting process resembles that of arthropods and other ecdysozoans, organisms that grow by periodically shedding external coverings. Before molting, tardigrades develop a new cuticle beneath the old one, then split and crawl out of the outgrown covering. The shed cuticle often remains intact, appearing as a ghostly empty shell that still resembles the tardigrade that abandoned it. Under microscopes, these molts can be mistaken for actual individuals until observers notice they're hollow and immobile. Some species lay eggs inside these shed cuticles, repurposing them as protective egg cases. Molting frequency depends on growth rate, food availability, and environmental conditions. Well-fed tardigrades in favorable conditions may molt frequently as they grow rapidly toward reproductive maturity. Those in poor conditions may extend intervals between molts or cease growing entirely until circumstances improve. The lifespan of tardigrades varies considerably between species and environmental conditions. In laboratory cultures with optimal conditions, some species live several months to over a year in active states. However, when accounting for time spent in cryptobiosis, individual tardigrades might persist for many years or potentially even decades. Calculating true lifespan becomes complicated when organisms can suspend metabolism for extended periods. Should dormant time count toward age if biological processes are essentially paused? This philosophical question aside, the practical longevity of tardigrades, their ability to persist across time regardless of metabolic state, exceeds that of most similarly sized organisms by enormous margins. Some researchers hypothesize that particularly long-lived individuals might accumulate centuries of existence when combining brief active periods with extended dormancy. While unproven, this possibility remains theoretically plausible given documented cases of multi-decade dormancy. If true, individual tardigrades might witness environmental changes across timescales usually associated with species evolution rather than individual lifespans. Tardigrades reproduce through both sexual and asexual means depending on species and circumstances. Sexual reproduction involves males transferring sperm to females, typically during molting periods when the female's cuticle is soft. Fertilization may occur internally or externally depending on species, with eggs subsequently being laid into the environment or inside shed cuticles. Asexual reproduction through parthenogenesis occurs in some species, where females produce viable eggs without fertilization by males. This strategy allows rapid population growth when conditions are favorable and colonization of new habitats by single individuals. Parthenogenetic populations may persist for many generations without males appearing. The flexibility to switch between reproductive modes provides adaptive advantages. When population densities are low and finding mates is difficult, parthenogenesis allows reproduction to continue. When populations are denser and genetic diversity becomes advantageous, sexual reproduction increases genetic variation that may improve adaptation to changing conditions. Embryonic development in tardigrades occurs inside eggs, with development rates depending heavily on temperature and moisture. In favorable conditions, eggs may hatch within days. Under cooler temperatures or variable moisture, development slows considerably or may pause entirely until conditions improve. Inside eggs, embryos develop through characteristic stages visible under microscopes in species with transparent eggshells. Cell division proceeds, organs form, and the miniature tardigrade gradually takes shape. By the time of hatching, the juvenile possesses all adult structures in functional form, ready to begin independent life immediately. Egg survival capabilities often match or exceed those of adult tunstates. Eggs can withstand desiccation, freezing, and other extremes while protecting developing embryos inside. This ensures that even if adult populations are eliminated by catastrophic conditions, eggs may survive to repopulate habitats when favorable conditions return. Some tardigrade populations show distinct seasonal patterns in abundance and activity. In temperate regions, populations may peak during spring and autumn when moisture and moderate temperatures create optimal conditions. During hot, dry summers or frozen winters, populations largely enter cryptobiosis, becoming virtually undetectable until favorable seasons return. These seasonal cycles create boom and bust population dynamics. During favorable periods, tardigrades emerge from dormancy, feed intensively, reproduce rapidly, and populations explode. As conditions deteriorate, most individuals enter tun states, and active populations crash to near zero before the cycle repeats. Understanding these patterns helps researchers locate and study tardigrades more effectively. Sampling during population peaks yields far more specimens than sampling during dormant periods. It also reveals how tardigrade communities contribute to ecosystem functions during active periods, while essentially withdrawing from ecosystems during dormancy. In tropical regions with more stable year-round conditions, seasonal patterns may be less pronounced. Instead, populations might fluctuate with rainfall patterns, showing activity during wet periods and dormancy during occasional dry spells. This creates different population dynamics compared to temperate species experiencing predictable seasonal cycles. Tardigrades have become important model organisms for studying extremophile biology and cryptobiosis. Their ability to survive conditions lethal to most life makes them valuable for understanding biological limits and adaptations to extreme environments. Research laboratories worldwide maintain tardigrade cultures for ongoing investigations into their remarkable capabilities. Their small size, short generation times, and ease of culture make them practical for laboratory research. Unlike larger organisms requiring extensive facilities, tardigrade colonies can be maintained in simple petri dishes with moss or algae cultures. This accessibility enables researchers with modest budgets to conduct sophisticated studies on extreme survival mechanisms. Findings from tardigrade research often have applications extending far beyond understanding these specific organisms. Their protective proteins, DNA repair mechanisms, and cryptobiotic strategies inspire developments in medicine, agriculture, biotechnology, and space exploration. They serve as living libraries of evolutionary solutions to problems humans also face. The DNA repair mechanisms in tardigrades have attracted intense research interest. Their cells can tolerate massive DNA damage from radiation or desiccation, then repair it efficiently when conditions normalize. Understanding these repair systems could lead to better treatments for radiation exposure, improved cancer therapies, or enhanced preservation of biological materials. Researchers have identified specific proteins unique to tardigrades that protect DNA during stress. When expressed in other organisms experimentally, these proteins confer some protective benefits, suggesting they could be adapted for practical applications. Imagine pharmaceutical treatments incorporating tardigrade-inspired proteins to protect patients undergoing radiation therapy or to preserve transplant organs longer. The DSUP protein mentioned earlier has been successfully expressed in human cells in laboratory experiments, where it reduced DNA damage from X-rays. This proof of concept demonstrates that tardigrade protective mechanisms can function in completely different biological contexts, opening possibilities for biomimetic applications borrowing nature's solutions to engineering and medical challenges. Tardigrades have traveled to space multiple times since the groundbreaking 2007 experiment. Subsequent missions have further explored their space survival capabilities, testing longer exposures and different conditions. Some experiments examined whether tardigrades could complete entire life cycles in space, including reproduction and development. Results have been remarkable. Not only do tardigrades survive space exposure, but some individuals successfully reproduced afterward, producing viable offspring. This suggests that space exposure doesn't necessarily cause permanent reproductive damage. These findings have implications for understanding how life might persist during interplanetary travel or on other worlds. Space agencies view tardigrade research as relevant to long-duration space missions. If we understand how tardigrades protect themselves from cosmic radiation and vacuum, we might develop better protection for astronauts or design biological systems capable of surviving Mars conditions or long journeys between planets. In 2019, an Israeli lunar lander called Bereshit crashed on the Moon's surface. Among its cargo were thousands of dehydrated tardigrades in ton states. The crash scattered these organisms across the lunar landing site, raising the intriguing possibility that tardigrades currently exist on the Moon, dormant but potentially still viable. The Moon's environment is extraordinarily hostile. No atmosphere, extreme temperature swings, intense radiation, and complete absence of water. However, these conditions aren't necessarily lethal to cryptobiotic tardigrades. In their ton state, they might persist on the lunar surface for years or even longer, waiting indefinitely for water that will never arrive. This accidental release sparked debates about planetary protection and contamination of celestial bodies. While tardigrades cannot reproduce or become active on the waterless moon, their presence raises questions about whether we should allow even dormant Earth organisms on other worlds. The incident highlighted need for stricter protocols governing biological materials on space missions. Scientists estimate the crashed tardigrades could potentially survive on the moon for years in their dormant state. However, without water to rehydrate them, they'll never reactivate. They exist in a state of suspended animation on an alien world, neither alive in any functional sense nor truly dead, frozen in biological stases on the lunar surface. This peculiar situation makes them perhaps the most distant terrestrial organisms ever, residing on another celestial body despite never having evolved there. Future lunar missions might even recover samples from the crash site to test whether the tardigrades remain viable after extended lunar exposure, providing data on long-term cryptobiosis limits. The thought of Earth's toughest microscopic survivors scattered across the moon feels both scientifically significant and oddly poetic. They represent life's resilience carried beyond our planet, dormant ambassadors waiting in lunar dust for conditions that will never come. Tardigrade research has inspired biotechnology applications in preservation of biological materials. Their protective proteins and cryptobiotic mechanisms offer models for stabilizing vaccines, preserving donor organs, or protecting probiotics during storage. Companies are exploring whether tardigrade-inspired technologies could improve storage of temperature-sensitive pharmaceuticals. Some researchers investigate whether tardigrade protective strategies could help preserve crops during droughts. If plants could be engineered to produce similar protective proteins, they might survive water shortages better, potentially improving agricultural resilience in drought-prone regions. While technically challenging, the concept demonstrates how understanding tardigrade biology could benefit human food security. The cosmetics industry has even explored tardigrade-derived compounds for protective skincare products. Marketing claims aside, the underlying science suggests that molecules protecting tardigrades from UV radiation and oxidative stress might offer similar benefits in topical applications. This represents an unusual intersection of extremophile biology and consumer products. Educational programs worldwide use tardigrades to engage students in microscopy and biology. Their combination of accessibility, found easily in local moss, and remarkable characteristics makes them perfect subjects for student investigations. Many students remember their first glimpse of a living tardigrade as a transformative moment that sparked interest in science. Classroom activities involving tardigrade collection and observation teach practical skills in microscopy, species identification, and ecological sampling while maintaining student engagement through the inherent fascination these organisms inspire. Teachers appreciate that tardigrade projects require minimal expensive equipment, yet deliver maximum educational impact. Citizen science projects have enlisted public participation in documenting tardigrade distribution and diversity. Volunteers collect samples from the local areas, photograph specimens, and contribute observations to databases helping scientists map species distributions globally. These projects democratize scientific research while expanding data collection beyond what professional researchers could accomplish alone. Tardigrades have appeared in popular culture, ranging from comic books to television shows. Their resilience and alien appearance make them appealing characters for science fiction narratives. Some portrayals exaggerate their abilities humorously, depicting them as nearly invincible creatures, while others use them as inspiration for alien life forms. This cultural presence raises public awareness of microscopic biodiversity and extremophile biology. When people encounter tardigrades in entertainment media, they often seek additional information, discovering the real science behind the fictional portrayals. This creates opportunities for science communication and public engagement with research. The tardigrade effect in science communication refers to using charismatic microscopic organisms to attract attention to broader biological concepts. Their combination of cuteness and toughness makes them unusually effective ambassadors for science education, reaching audiences who might not otherwise engage with microscopic ecology or evolutionary biology. Museums and science centers worldwide feature tardigrade exhibits, often including live specimens under microscopes for visitor observation. These displays reliably attract crowds, with people fascinated to observe real organisms they've heard described as indestructible or able to survive in space. Interactive elements might allow visitors to simulate extreme conditions, learning how tardigrades would respond. Such exhibits effectively communicate scientific concepts including adaptation, evolution, extremophiles, and biodiversity at microscopic scales. They demonstrate that extraordinary biology exists not only in exotic locations but in the everyday environments surrounding us. This accessibility helps break down barriers between public audiences and scientific knowledge. Gift shops at these institutions often stock tardigrade merchandise, plush toys, t-shirts, posters, and books. This commercialization, while perhaps seeming trivial, actually extends science communication into daily life. A child wearing a tardigrade shirt might explain to friends what makes these organisms special, essentially becoming a science communicator themselves. Tardigrades symbolize resilience in unexpected places. They remind us that toughness doesn't require size, strength doesn't demand visibility, and survival doesn't necessitate complexity. Their existence challenges assumptions about life's fragility, demonstrating that with proper adaptations, organisms can endure conditions once thought absolutely incompatible with biology. This symbolic power extends beyond science into broader cultural meanings. People facing challenges often reference tardigrades as inspiration. If microscopic creatures can survive space, extreme temperatures, and decades without water, perhaps human difficulties become more manageable by comparison. While anthropomorphizing organisms risks biological inaccuracy, the metaphorical value remains genuine. Tardigrades also symbolize the hidden complexity of nature. Beneath our notice, entire worlds of remarkable organisms pursue their lives, facing challenges and evolving solutions we are only beginning to understand. They represent humility before nature's diversity, reminding us that significance doesn't correlate with size or visibility. The study of tardigrades demonstrates how much remains unknown even about Earth's biology. Despite centuries of scientific investigation, we continue discovering new species, identifying novel survival mechanisms, and uncovering unexpected capabilities. Each new finding generates additional questions, expanding rather than closing the frontier of knowledge. This ongoing discovery process excites researchers because it suggests tardigrade biology still holds secrets waiting to be revealed. Perhaps additional protective proteins await identification, novel metabolic pathways remain uncharacterized, or undiscovered species possess capabilities exceeding those of currently known varieties. The future of tardigrade research seems boundless. For science as a whole, tardigrades illustrate how studying seemingly obscure organisms can yield insights with broad applications. Research motivated purely by curiosity about how these creatures survive extremes has generated practical applications in medicine, biotechnology, and space exploration. This justifies basic research without immediate practical goals. We cannot predict which fundamental discoveries will eventually transform technology or society. Tardigrades may hold keys to understanding how life might exist beyond Earth. Their ability to survive space conditions suggests that organisms could potentially travel between planets on meteorites, a concept called panspermia. While speculative, this idea proposes that life might spread through the cosmos on rocky fragments blasted from planetary surfaces by impacts. If tardigrades can survive years in space and impact shockwaves, might similar organisms exist elsewhere in our solar system or beyond? Could life have originated on one world and spread to others through natural processes? These questions remain unanswered, but tardigrades prove that such scenarios aren't impossible from a biological perspective. Astrobiologists study tardigrades to define the environmental limits compatible with life. Understanding their maximum tolerances helps identify potentially habitable zones on other worlds and guide searches for extraterrestrial life. If life can adapt to such extremes on Earth, perhaps similar adaptations allow survival in Martian soil, beneath Europa's ice, or in other seemingly hostile environments. Their existence also reminds us that habitable is a flexible concept. Environments we consider completely uninhabitable might support organisms with appropriate adaptations. This expands our conception of where life might exist, both on Earth and on distant worlds we've barely begun to explore. As we conclude this journey through 100 facts about tardigrades, consider what we've learned. These microscopic creatures survive in space, endure temperatures from near absolute zero to above boiling, withstand crushing pressures and sterilizing radiation, persist through decades of complete desiccation, and carry evolutionary lineages stretching back over 500 million years. They inhabit every continent, thrive in environments from mountaintops to ocean trenches, walk on 8 stubby legs with distinctive bear-like gaits, reproduce through elaborate ornamented eggs, and possess unique proteins found nowhere else in nature. They've inspired scientific research, medical applications, space exploration strategies, and captured public imagination worldwide. Yet perhaps their greatest lesson isn't about their specific superpowers, but about patience and persistence. Tardigrades don't fight against harsh conditions, they wait them out. They don't demand perfect environments, they endure imperfect ones. They don't require constant activity, they know when to pause and when to proceed. In our own lives, there's wisdom in this approach. Not every challenge requires immediate action. Sometimes survival means recognizing when to conserve energy, when to seek shelter, when to pause and wait for better conditions. The tardigrade strategy of cryptobiosis, temporarily suspending normal life to preserve future possibilities, offers a metaphor for human resilience. As you drift towards sleep tonight, imagine yourself with the patient endurance of a water bear. Picture yourself capable of weathering any storm by simply waiting it out, protected by invisible shields of resilience. Envision yourself small enough to slip through the cracks of impossibility, tough enough to survive what seems unsurvivable. These tiny creatures have walked the earth for over 500 million years, witnessing the rise and fall of countless species, enduring every catastrophe our planet has suffered. Their presence today, essentially unchanged, testifies to the power of adaptation, the value of flexibility, and the triumph of patience over time. In moss cushions near your home, in the soil of your garden, perhaps in the gutters of your roof, tardigrades continue their ancient existence. They feed, reproduce, occasionally face challenges, enter their tun states, and eventually emerge when conditions improve. They neither hurry nor hesitate, they simply persist. May their story bring you comfort as you settle into rest. May their resilience remind you of your own strength. May their patience inspire your own approach to challenges. And may their persistence across deep time help you maintain perspective on your own brief moment in the vast flow of existence.